standing room only, so we hope those were enjoyable. And I want to remind you again that you can email your questions to nobelconference@gustavus.edu. And if you post on social media, please put hashtag Nobel Conference. We'd love that. And now it's my privilege to welcome to the stage President Volin, Provost Kelly, and Professor Anandino, as well as Dr. Carolyn Bertozzi, to do our next talk.
Today, Gustavus Adolphus College honors Dr. Carolyn Bertozzi. Dr. Bertozzi serves as the Ann T. and Robert M. Bass Professor of Chemistry at Stanford University and as the Baker Family Director of the ChEM-H Institute at Stanford. The institute combines chemists, engineers, biologists, and clinicians to study life at the molecular level and apply it to improving human health. Together with Barry Sharpless and Morton Meldal, she was awarded the Nobel Prize in Chemistry in 2022. Dr. Bertozzi graduated summa cum laude with an undergraduate degree in chemistry from Harvard University. She then earned her PhD from the University of California, Berkeley, where her research focused on the synthesis of carbohydrate analogs for biological studies. Following this, she completed a postdoctoral fellowship in cellular immunology at the University of California, San Francisco, and held a faculty appointment at UC Berkeley before joining the faculty at Stanford University in 2015. Dr. Bertozzi is renowned as a pioneer in the field of bioorthogonal chemistry, a term she coined. Bioorthogonal chemical reactions occur inside living systems without interfering with native biochemical processes. Her innovative copper-free click chemistry method provided a way for researchers to visualize and study biomolecules in their native environments without the use of toxic catalysts. This groundbreaking research revolutionized the way that scientists observed and conceptualized cellular processes in real time and allowed researchers to attach probes to specific molecules in living cells. Dr. Bertozzi's work bridges fundamental chemistry with practical medical applications, and the unprecedented visualization of biological processes that she and members of her laboratory developed has led to major innovations in diagnostics, immunotherapy, and drug delivery systems, particularly in the treatment of cancer patients. In addition to receiving a Nobel Prize in Chemistry in 2022, Dr. Bertozzi has received accolades, including being inducted into the National Inventors Hall of Fame in 2017, and numerous awards, including the MacArthur Genius Award in 1999, the AAAS Lifetime Mentor Award in 2022, the President's Innovator Award through the Society of Glycobiology in 2020, and the Priestley Medal, the highest honor bestowed by the American Chemical Society in 2024. She is a member of the National Academy of Sciences, the National Academy of Medicine, the National Academy of Engineering, and the American Academy of Arts and Sciences. Dr. Bertozzi has also served as a mentor to over 100 doctoral and postdoctoral researchers who now lead their own research programs. And she has been a prominent advocate for the inclusion of underrepresented groups in the sciences. In fact, the principles of her research lab begin with these words, quote, "In the Bertozzi lab, we are committed to performing basic research and developing new technologies that serve humanity. Our endeavors are supported by a diverse and inclusive working environment We welcome researchers of all backgrounds and perspectives. For these contributions, Gustavus Adolphus College is both honored and privileged to bestow an honorary degree upon Dr. Carolyn Bertozzi. Upon recommendation of the faculty, Dr. Bertozzi is presented with the degree of Doctor of Science honoris causa.
For your unwavering efforts to understand, visualize, and inform the world about real-time cellular and chemical processes, Gustavus Adolphus College is privileged to bestow an honorary degree upon you, Dr. Bertozzi. Now, by the virtue of the authority vested in me by the Board of Trustees of Gustavus Adolphus College, and upon recommendation of the faculty and approval of the Board of Trustees, I hereby confer upon you, Carolyn Bertozzi, the degree of Doctor of Science honoris causa with all the rights, privileges, and honors pertaining thereto.
Okay. Okay.
We're going through this row.
We will welcome Dr. Bertozzi's comments as soon as we get organized up here.
Okay. Thank you. What an enormous honor it is for me to be here among you under these circumstances. Let me just express my deepest gratitude to your president, your provost, your distinguished faculty, and mostly to the students who took so much time from their lives, several of which are handling me, while I'm here. Thank you, liaisons, for that hard work that you do. I don't think you've had to implant a tracker into my body quite yet. I haven't gone too far astray, but it's such an incredible honor for me to be part of this community with so many brilliant students interested in all different dimensions of scholarship. And it's really wonderful for me to have the experience of participating in this conference where I can learn so much about, in this case, the subject of sugars. This is an area of biology that I have been interested in ever since my very first days as a PhD student. And the work that you alluded to in this very kind introduction on this brand of chemistry called bioorthogonal chemistry, that was the subject that the Nobel Committee recognized me for. But actually, all of that chemistry was really just a means to another end, which was to study sugars and the biology of sugars. So that really is the focus of my laboratory, and that is what I'm going to talk about today. So you've already heard about sugar from the talks this morning, and you'll hear certainly more over the next day or so. And when a person hears the word sugar, the first thing you think about is what you eat, right? The sugar that you eat and the effect that it has on your physiology. And so far, that has been the focus of this conference. But I'm here to tell you about a different dimension to the biology of sugar molecules. Not only do we eat simple sugars and use them for energy, but we also have sugars that coat the surface of all of our cells. And this is true for basically all organisms on the planet Earth. It's true for us, it's true for all the animals, it's true for bacteria, it's true for plants, in fact. They have a cell wall that's made up of sugars. And I'm going to talk about why it is that we have the sugar coating, how the sugar coating can turn bad and contribute to diseases, in particular cancer. And then finally, some ideas that we have in my lab for developing cancer therapies that basically interfere with this disease biology. Okay, so here we are. I thought since we had donuts for breakfast, we could have M&Ms after lunch. Everyone would recognize what these are. When I took biology in college, so this would have been, mid-1980s, that was the first time that I learned from our professor that our cells are coated with sugars, and my biology professor drew the analogy to the peanut M&M. And he said, if you cut through the middle of a peanut M&M, you would see this, and this is kind of like our cells. There's a sugar coating. Then, in our cells, we have this layer of fat called the lipid or plasma membrane. That's like the chocolate. And then there's all these proteins and other things, and that's like the peanut, okay? And somebody raised their hand in that classroom and asked, well, what's the purpose of the sugar coating on our cells? And back in the mid-1980s, our professor said, well, people don't really know, but maybe it's the same as the function of the sugar coating on the peanut M&M. And people of a certain age will remember the commercial on TV because of that sugar coating, M&Ms melt in your, but not in your hand. Right? It's a protective shell, okay? It turns out it's much more complicated and much more interesting than that. So as time went on, and again, decades unfolded, it became clear that the structures, the chemical composition of the sugars on the surface of our cell, those structures are much more complex than the simple sugars that we eat, the glucose, the fructose we learned about this morning, the sucrose. These are very complex carbohydrates, people would call them. And nowadays, we more often call them glycans. And by the way, the prefix glyco is a Greek prefix for sugar. Okay, so glycans, just another word for complex carbohydrates. I think a better metaphor for these sugars and their structures and what they look like, it's not so much the peanut M&M, it's more like the surface of the planet Earth, where if you had a drone flying across the surface of the planet, you would see something like this, where there's trees and grass and shrubs, there's small trees and large trees, and there are features on the planet Earth. And that's what the surface of our cells look like, where the trees and the grass and all of the vegetation is complex glycans. And they move around. And here's a video, which is actually a computational model generated by computer graphics, in which you can kind of predict how these complex glycans will behave. And it's almost like if you've ever been to one of these giant kelp forests in an aquarium, where these long, tall, kelp trees are basically swaying in the water. And that's how I picture these complex glycans on the surface of our cells. And once it was clear that the structures are very complicated, and once the technologies were available to really get into the weeds of what those structures look like, it became clear that this coding of complex glycans is a language. It is a way that the cell can communicate to the outside world about its status. And we now know that different cell types have different patterns of these glycans. And these are just some cartoons to illustrate what we imagine they might look like. Tall trees, long strings of beads, little chunks of grass and shrubs, all kinds of different features, but it's not random. So the pattern of all of these different kinds of molecules on a cell from your muscle tissue will be distinct from the pattern on cells from your brain. And these patterns emerge as organs develop during embryogenesis. So that's a hint that there's something much more complicated and that these sugars are actually contributing to the complexity of multicellular organisms. So I became interested in this science really my very first year of graduate school. And I studied the chemistry of sugars as a PhD student. And then later, I took a postdoctoral position and started to learn more about the contribution of sugars in human diseases. And as I transitioned over to the more biological side, I started to understand that discoveries in cell surface sugar biology have already really transformed the field of medicine. For example, one of the things that those cell surface sugars are trying to tell you is your blood type. Your blood type is determined by the structures of your cell surface sugars. So how many people, show of hands, are blood type O? And so am I. And so what we all share in common is we have this chemical structure. For any chemist in the room, you'll be able to understand what these different lines and symbols mean. That is a glycan that is found on your blood cells, built within the cells by a pathway of enzymes. And by definition, if you have that structure, you are blood type O. How many people are blood type A? Quite a few, all right. You people have an enzyme that I don't have, and you are able to put that red building block, that's a simple sugar, onto the underlying structure to make a new structure, which is blood type A. And then there's a slightly different blood type, which is B. Who's the Bs? This is usually more rare, at least in the Midwest. And you're like, hey, represent. There's nothing to be ashamed of, to have blood type B. So you're B, and then some people are AB. Yeah, ooh, you're special. Because you got the gene for A from one parent, the gene for B from the other parent, so you have a 50-50 mixture, okay? This was a really important discovery because once we figured out how to determine a blood type, we could then determine how to give you a safe blood transfusion. If you didn't know your blood type and you just took a blood donation from anybody in the room, the chances are you wouldn't get a matched blood type and your body would reject it. And so understanding the blood type biology was really important to save people's lives with blood transfusions, but it also taught the world of biology and medicine about the potential importance of cell surface sugars in the immune system in particular, okay, which is now going to be relevant. Because, the font's got a little funky here, sorry about that. Where I got interested in these cell surface sugars was in the context of cancer. Now, it's been known for about half a century that if you take an inventory of the structures of the glycans on healthy cells and compare that to the pattern on a tumor that develops from those cells, there are some pretty dramatic changes in the composition of those cell surface sugars. We have known this now, like I said, for half a century. But until recently, people really did not have an understanding as to why this might occur. And so when I became a new professor back in 1996 at UC Berkeley at that time, it was the goal of my young lab to try to figure out why. Why did these changes occur? What does it mean for the cancer to have different sugars? And if that's important in the disease, maybe there would be an opportunity to develop a new kind of cancer medicine, Okay, so that was the thought process back in 1996, which now, that's almost 30 years ago. Time has really flown. And where we started was with one particular phenotype, as we call it, that's quite common amongst cancers. So for many human cancers, if you look at the sugars on the healthy versus the cancer cell, you will see that the cancer is overproducing a particular type of sugar, which we call sialic acid, in pink here. Don't get distracted by the word acid in sialic acid. There's nothing corrosive about it. It's a sugar, okay? And we have, turns out, 9 of these simple sugars, and this is one of them, okay? So your healthy cells all have what I like to think of as a well-manicured garden of sialic acid. Here's the chemical structure for the chemists. One thing I've learned in my 30 years doing this is biologists are okay with the Lucky Charms pictures, the little symbols with the little colors, you know, the diamonds and the squares and the circles. Chemists, I can see them getting more and more agitated as the minutes tick by. They're like, well, what does it look like? That's what it looks like, okay? So that well-manicured garden on your healthy cells becomes like a tropical jungle on the cancer cells. This observation dates back to the 1980s, but until recently, nobody understood why this might happen. And what we discovered in my lab at Berkeley at the time was that it has to do with the interaction between the cancer cells and our immune system. So now let me give you a little bit of a primer on cancer immunology. How many people have heard of cancer immune therapy? Is this now like a household term? Okay, I think a lot of people have heard that term. Immune therapy for cancer is probably the biggest breakthrough in oncology of my lifetime. It has really transformed the practice of how we treat cancer patients. So I think it's important to understand the basics of this. In case you didn't know, your immune system is protecting you from cancer most of the time. Right now, in this audience, your immune system is protecting you from cancer. And that's a great thing because most of us will live most of our lives cancer-free, okay? And the way that our immune system helps us out is you have immune cells. Those are your white blood cells. They're coursing through your bloodstream all the time. Their job is to patrol your body and look either for foreign invaders, like bacteria or viruses, or to look for your own cells that have gone bad, cancer cells, for example. And the way they do this is they will literally park themselves onto each of your cells one at a time and sample the merchandise. They're literally tasting your cells. So in this particular graphic, the green cells are immune cells. They're actually a subset of immune cells that are called T cells. And the giant purple cell is a cancer cell. And you can see that these immune cells have latched on. They form what we call an immune synapse, where the two cells are touching each other. And those immune cells are just sampling the molecules on the surface of the cancer cell. And if they sense that something is not right, they will respond, and they will kill that cell. So how do they make that decision? How do they know, just from tasting the surface, whether something has gone wrong in that cell? Well, those immune cells, and now this is a blow-up, cartoon blow-up of that synapse, the immune cells have proteins anchored to the surface of the cell. We call them receptors. Some of those receptors are activating receptors. That's the green shape. Those activating receptors are looking for molecules on that cancer cell that would be an indicator that there's something wrong with it. And we have a built-in system for this. If one of your cells has been mutated, let's say you were lying out in the sun for too long, your skin cells absorbed the sunlight, it damaged your DNA. That's why we wear sunscreen. But if you have enough of that damage in your DNA, the cell is like, I don't feel good, and it'll show signs of that on the surface. And your immune cells will come along and be like, nope, get rid of it, before it turns into a cancer, okay? That's what the activating receptors are for. So if the receptor binds to one of those damaged signals, and it's like a lock and key, like a handshake type of interaction, that triggers a pathway inside the immune cell, which leads to a bomb drop. That's supposed to say bombs away there, okay? And that's the end of the cancer cell. Now, this is a dangerous superpower that your immune cells have. Because what if they make a mistake? What if they think that there's damage, and they say, great, let's start killing. Kill that cell, kill that one, kill that one, kill that one. What if they accidentally start killing your healthy, normal cells? That would lead to autoimmune disease. So who here has heard of rheumatoid arthritis? Okay. Who's heard of psoriasis? Okay. Lupus. Multiple sclerosis. Yeah. These are common autoimmune diseases where the immune cells are killing your own healthy cells. They have made a mistake. Now, fortunately, most of us don't have autoimmune diseases. You know, some people are not so fortunate. For those of us who don't have autoimmune reactivity, it's because there are these other receptors on immune cells, the inhibitory receptors. Some people call them checkpoint receptors. Their job is to look for different kinds of molecules on the surface of a cell. Because our healthy cells should have healthy signals, and those healthy signals do a handshake with the inhibitory receptor, and that blocks or inhibits the ability of the immune cell to drop the bomb. So when everything is working well, healthy cells have healthy signals, immune cells go to sleep. Cancer cells have damaged signals, immune cells drop bombs. Okay, that's the perfect world. Cancers are smart and sneaky and very good at becoming resistant to things that try to kill them. So every once in a while, a cancer cell will figure out how to put a healthy signal on the surface and trick the immune system. Okay, so all of this biology was discovered and worked out kind of in the first decade of the 2000s, and that ultimately led to a new type of medicine, these immune therapies. So what the first generation of immune therapies do is they block the handshake between inhibitory receptors and the healthy signals. They block that interaction so that the cancer cell cannot inhibit the immune cell, and instead, the damaged signal does its job, provokes the immune cell to kill the cancer cell, okay? So they block an inhibitory receptor. This was a huge breakthrough because all of a sudden, certain types of cancers, which were previously 100% fatal, could suddenly be cured for some patients. And we don't use the word cure and cancer in the same sentence very often. It's very hard to cure most cancers. But immune therapies sometimes can do it. It was a really big deal when some of the first clinical trial data was released back, this would have taken us back to around 2011 or 2012. And the clinical trial data typically looked something like this. These are curves, we call them Kaplan-Meier curves. But what this is, you'd have a bunch of patients, hundreds of patients, in a clinical trial, and you would just look at their survival as a function of time on different therapies. And what people found is that conventional chemotherapies might extend somebody's life, but they really didn't cure anybody. And eventually, all the patients would die. One of the very first indications where immune therapies were tested was in metastatic melanoma, that skin cancer that has spread elsewhere in the body. That was 100% fatal, and yet maybe 30 to 40% of patients with that diagnosis would get on these immune therapies, and they would live and live and live and live. And so you see the immunotherapy curve just flattens out. Those patients are cured, but not all of them, maybe 20 or 30%, depending on the clinical trial, okay? But even to have 30% of patients cured was nothing short of miraculous. This was a huge breakthrough, and basically now, fast forward another 14 years or so, every pharmaceutical company that aspires to treat cancer patients will have one of these types of immune therapy drugs in its portfolio. And by the way, all of those drugs are what we call biologic drugs. They're monoclonal antibodies, or they're protein drugs that are administered by an infusion. And they're all pretty much the same, but these are among the most important new types of cancer drugs that are being used today. One of the people who was cured of his metastatic melanoma was our former president, Jimmy Carter. Do you remember that? In 2013, he got into the clinical trial for Merck's immune therapy, and he was one of the lucky 30% or so of patients who was cured of his disease. And at the time, I think he was about 91 years old. Now, he lived another decade. So, that's an example of what can happen with these kinds of drugs. But it's not perfect because, if you're not in the lucky 20 to 30%, you don't benefit from these drugs. And here are some numbers just to give you a sense of the survival or the cure rate for different types of cancers when patients are treated with these drugs. Melanoma is one of the most responsive types of cancer. And nowadays, people have improved the drugs and the protocols, so maybe almost half of those patients might have a durable remission. But these other types of cancers, for which immune therapies have been approved by the FDA, have a much lower response rate, okay? So we still cannot cure most people with these other cancers, and then the cancers not on this list, many of them really haven't responded at all. Lots of cancers have been tested in the clinic. Not all of them will respond. So the big question in this field has been, why doesn't everybody respond? What's going on in those patients that are not responding? And maybe the targets of that first generation of immune therapies are not important in every patient, and maybe for some patients there are other biological mechanisms by which the cancer cell can inhibit the activity of the immune cell that's attached to it, okay? So what are we missing? Well, we speculated in my group that we were missing the cell surface sugars. Because remember, 50 years ago, people who characterized these structures discovered this common cancer phenotype where there's an overproduction, a tropical jungle of sialic acids on the cell surface. Well, fast forward into the year 2010, and we discovered that there are receptors on immune cells that will bind that sugar, sialic acid. And here's some technical language. One thing scientists are really good at is creating acronyms that nobody understands or can pronounce, and we are among them in this regard. This is a cartoon illustrating a family of immune cell surface receptors that are called the sialic acid binding, Ig-like lectins. So all that packaged up, we call them the Siglec receptors for short. And the way the cartoon is drawn, the parts above the yellow line, those parts are outside the cell, on the surface of the cell, on the immune cell. And you can see that some of them are tall, some of them are short. They have all these different blue shapes and components. But what they all do is bind that sugar, sialic acid. Then there's the yellow line. That's the cell surface. That's the membrane of the cell. And then the parts below the yellow line are inside the cell. And different members of this family are found on different types of immune cells. So by the way, we have, a dozen or so different types of immune cells. I like to think of your immune cells like the military-industrial complex of your body's defense system. So just like for us as a nation, we might call upon the Army, the Navy, the Air Force, the Marines, the Coast Guard, depending on where the problem lies. Your immune system is the same way, but instead of Army, Navy, Marines, and so on, we have T cells, B cells, macrophages, NK cells. But all of them contribute. And they all have members of this family. And the reason we thought these would be interesting receptors to look at is because there's these parts inside the cell, the red brick, the purple brick. Those are bricks that are also found in one of the famous checkpoint receptors against which we have these first generation of drugs. It's a receptor called PD-1 on the T cell. And those parts that are inside the cell, the red part and the purple part, some of these Siglec receptors have the exact same bricks as that T cell checkpoint receptor, PD-1. And so we sort of analogized that, just as in the conventional cancer immune interaction against which that first generation of drugs was made, you picture the checkpoint receptor on the T cell interacting with something on that target cell, and that stuff that's inside the cell, it's got the same stuff over here. So we thought, well, maybe the reason so many cancers have the tropical jungle of sialic acid is because all of that sialic acid allows those cells to grab onto those Siglec receptors, which could be another way to put an immune cell to sleep. And maybe the patients that have the highest amounts of sialic acid, maybe those are the patients that are not responding to that first generation of checkpoint inhibitor drugs, because maybe for those patients, those Siglec receptors are just more important or making a bigger contribution to the immune suppression than is PD-1. So we wanted to test that hypothesis experimentally, and we spent about 5 years looking at patient biopsy samples, looking at how much sialic acid was there, comparing that to what we knew about their clinical responses to drug treatments. We set up animal models to look at cancers in the mouse and look at their sialic acids and how that relates to the immune system. And we finally came to the conclusion that the sialic acid Siglec biology might even be more important than the PD-1 or checkpoint biology. And we should really think about trying to make immune therapies that target the biology on the right, either on its own or maybe together in combination with those checkpoint inhibitors for the biology on the left. Okay, so in a nutshell, this is what we learned from that first decade of research on this subject. Cancer cells, when things are going well, should present a bad taste to immune cells. And when they taste bad, the immune cell gets rid of them. But if the cancer cell is coated with sialic acids at a high density, because those sialic acids can grab onto the Siglec receptors, that basically delivers the message to the immune cell that, hey, I taste good. There's nothing wrong with me. Leave me alone. And a cancer cell that's able to shoo away immune cells is a cancer cell that can live another day, divide another day, grow into a tumor, spread throughout your body, and your immune system has been rendered helpless. How do you make a drug that turns this around? Okay, well, keeping with our kind of landscape metaphor, okay, we thought maybe we can make a medicine that behaves like a lawnmower, where we imagine parking the lawnmower on the cancer cell and mowing off the sialic acids. Just strip them right off the surface, and now that cancer cell doesn't have the capacity to grab on to Siglec receptors, and that should make them much more easily killed by our immune cells. So what does that lawnmower look like in terms of a molecular drug? What we did, and oh, again, sorry, the fonts got really weird here in the transfer. This is unreadable to you, but this is basically a protein that we made, which has two parts. The purple part is a monoclonal antibody that will park on cancer cells. It's been designed to interact with something that only cancer cells would have. It's called a tumor-specific antigen. And then the green part. The green part is an enzyme called a sialidase. It's an enzyme that will catalyze the hydrolysis of the linkage, or the cleavage of the linkage between the sialic acid sugar and what lies beneath. Okay? So again, the purple part parks, and the green part mows. And we need the purple part there because if we weren't parking the enzyme on the cancer cells, that enzyme might accidentally mow the wrong lawn. We don't want to mow your neighbor's lawn, or we want to mow your lawn, okay? And that's why we have to fuse that enzyme to that parking antibody. So how do we fuse these two proteins together, the green protein and the purple protein? We do it with bioorthogonal chemistry, okay? So this is the one slide where I will introduce the concept for which I won a Nobel Prize. What bioorthogonal chemistry allows us to do is to take biological molecules like proteins, which for the chemists in the room, you will recognize that a protein has a lot of different functional groups. And how many students here took organic chemistry class? Quite a few. And when you were taking that class, did you learn about protecting groups? Did you use them in your synthesis? Because you had to block certain functional groups so that they wouldn't get damaged by the chemistry you were doing. You protected them. Imagine if you had to do that kind of chemistry on something as large as a protein, with all of the functional groups that you would have to protect and block. It would be impossible. But bioorthogonal chemistry relieves you of that burden. These are chemical reactions that you can do even surrounded by biological molecules without any interference or interaction with the biological molecules. That's why it's orthogonal to biology. And so we developed two different bioorthogonal chemical reactions that we used to fuse the antibody together with the enzyme. And so for the chemists among us, there's the antibody in cartoon form. We figured out how to put aldehyde groups onto the antibody, which are orthogonal because there's no aldehydes normally on a protein. And then we developed a chemical reaction. We called it the Pictet-Spengler ligation. I won't go through why we did that, but we did. And that allows us to form a carbon-carbon bond between that organic molecule and the antibody. Now, if you can see, it might be too small from where you are, but right at the end here, there's that little group. Those are 3 nitrogens. Does anybody know what you call a functional group that has 3 nitrogens in a line? The azide. Yeah, that's an azide group. And we developed a reaction wherein the azide would click together with this kind of a structure, which is an 8-membered ring in the middle there called a cyclooctine. And the cyclooctine we attached to the enzyme, which is this green Pac-Man. So after putting the azide onto the antibody, we did the so-called copper-free click chemistry to fuse the proteins together, and that's how we made our first molecule of this type. And we tested antibody sialidase conjugates in a variety of different animal tumor models. This is one example. These are data from a breast cancer model in the mouse. It's a model that is known to be resistant to many other types of therapies. But what we found is typically we would grow tumors in these mice. We would graft the cancer cells into the mammary fat pad, so that's where a breast cancer would grow. And then at different time points, which is the arrows, the little arrows there, we would inject reagents into these animals and look at a change in the growth rate of the tumors, which is what the curve is reading out. And so when the animals were injected, either with just a saltwater solution, that's PBS, or with just the antibody with no enzyme on it, those tumors just grew and grew and grew. But if we had injected into the animals, in these two colors, it's the antibody sialidase fusion at 2 different doses, the tumors really didn't grow well at all. And when we harvested these tumors, what we found is there were no cancer cells left in them. They were just immune cells. Immune cells had come in there, did their job, killed off all the cancer cells, and you basically had a scar. And not only that, if we wait a month and try to grow another cancer in these same mice, the cancer doesn't grow at all because they've developed immunological memory against those cancer cells. They've been vaccinated against the cancer cells. So this was very encouraging, and we thought it was a good time to think about how to translate this discovery from the laboratory setting into more of like a human clinical development setting. And to do that, in my experience, it's best done in the hands of a company outside of academia. So we spun out a company called Paleon Pharmaceuticals. I don't know why I'm suddenly at the Harvard Chan School here. That was not on my original slide. So I don't know how that landed there, but I should be so lucky, but no, I'm not at Harvard. But Paleon actually is a company that's in the Boston area. It's in a town called Waltham, by the way. Okay, so we are in the neighborhood, but nothing to do with Harvard. And so our prototypes that we had made in my lab took the form on the left here, where we had a bacterial enzyme connected to that tumor-targeting antibody. But the scientists at Paleon kind of re-engineered the molecule to make it sort of more drug-like, is the term that they would use. Make it more friendly to manufacture on the large scale that you would need to do human clinical trials. And so they called this molecule their Eagle platform. And what they did is, instead of having 2 enzymes down at the bottom, they now have just 1 enzyme up at the top, which reduces the size of the molecule, and that's always helpful. And most importantly, instead of using a bacterial enzyme, as we had done in our prototype, they switched over to a human sialidase, which naturally is found in our bodies. And that's always helpful because humans don't like foreign proteins. We'll raise immune responses against them. So it's always nice to have a human protein as part of a human drug. So this molecule is now on its way into first human dose in probably mid of 2026. So hopefully by the end of next year, we'll have an early read on whether there's a there, and hopefully we'll be able to help more patients, either with this type of drug on its own or in combination with those first-generation immune therapies. And that's what we'll be following up on. Thank you. So if you want to know the outcome of the clinical trial study, you might have to invite me back in a few years. But anyways, for now, let me just close with a quick summary of the take-home messages. First, I told you, maybe for the first time, that all cells are coated with sugars. That might have been a revelation. Also, cancer cells have a different pattern than healthy cells, and they often have this dense overproduction of sialic acid. And nobody knew why that was until recently, and now we know that having those sialic acids allows the cancer cell to bind these Siglec receptors on immune cells, which put the immune cells to sleep. And that's of benefit to the cancer. And by understanding that, we were able to come up with a potential therapeutic intervention, which is to make these cancer-targeted lawnmowers that will strip the sialic acids off of the cancer cell and hopefully reveal to the immune system the true nature of the bad guy and allow the immune system to destroy it. And with that, thank you very much. I really appreciate it.
Hey, everybody.
So at this point, we're
I'm
gonna
Tane
set up
Danger.
again
I'm
for
Maggie
our
Kurth.
panel
We
discussion.
are here after, wow, what a lecture at the Nobel Conference at
Yeah.
Gustavus Adolphus College. This was our Nobel laureate who, this whole thing started, she got an honorary PhD from Gustavus, which is very cool. And then Dr. Bertozzi walked us through some of the most interesting science I feel like I've ever heard at a Nobel conference. I followed a lot of it. She had a lot of really good metaphors, which was very helpful for a person
Yes.
like me. But I don't know. Can you help us? Just she was giving a summary. just
Yeah,
now.
I mean, well, I love the fact that you were right about the candy coating.
Yeah.
Yeah, that's
if
exactly
people
the
were
same
watching,
analogy that she used.
I was just kind of riffing before this. I was like, she's gonna